Welcome back, everybody. Now that you know a little of the background information about photosynthesis, today I want to take a little bit more of an in-depth look at what happens in photosynthesis at the molecular level. Now, the first thing to understand is that photosynthesis actually takes place in two distinct steps, both of which happen inside the chloroplast. The first step is the absorption of light energy. And then that step produces molecules that are then used to produce high energy organic molecules such as glucose from carbon dioxide. The capturing of the light energy is often referred to as the light reactions. And these take place inside the thylakoid. They utilize the photosystems, which are molecular assemblages that include the chlorophyll. And each photosystem is associated with an electron transport chain. The electron transport chains are used to set up a hydrogen gradient, which then makes ATP. And a second electron transport chain makes a molecule called NADPH, which is very similar to the NADH that you already know about from cellular respiration. The creation of glucose from those products of the light reactions are often referred to as the dark reactions, and these occur in the stroma of the chloroplast, and they utilize the ATP and the NADPH that have been synthesized to make the glucose. And here's our picture of how it occurs. The light reactions are on the left side, and light and water in the chloroplast are used to make ATP, NADPH, and the waste product from water, oxygen. And those occur in the thylakoids, as I mentioned. And then in the stroma outside the thylakoids, carbon dioxide is added to an existing organic molecule in a process called the Calvin cycle. And the energy in ATP and NADPH is used to add this carbon dioxide and make an organic molecule. These molecules are eventually exported as glucose or sucrose. It doesn't matter which molecule is exported. We just need to know that a sugar molecule is going to leave the Calvin cycle. So let's take a look at the light reactions. As I said, they require sunlight, water, ADP, and NADP+. They release oxygen and they create ATP and NADPH where all the energy is now stored. And here's how it works. The chlorophyll captures a unit of light called a photon. This causes an electron to gain energy. We call it an excited electron. And then that is used to do both create a hydrogen gradient through a hydrogen pump and also to make NADPH. So the light energy has now been stored in these molecules. And this happens because the chlorophyll is in the thylakoid membrane and the electron is excited and then shuttled down a series of membrane proteins, in other words, down an electron transport chain, that slowly extract energy from the electron. There are actually two electron transport chains in photosynthesis, and we're going to see them in the next slide. You don't need to know the molecular details of the chain, but the general patterns of what happens. So let's take a look at that. So let's get situated in this diagram. Here we have the thylakoid membrane, and it's just a plasma membrane, as always, composed of phospholipids. Here we see what is called Photosystem II, which is a complex of many molecules, including chlorophyll. And then there is an electron transport chain of several proteins associated with the membrane, then a second photosystem, then some more electron transport proteins. And here we see ATP synthase. So let's figure out what's going on here. So light comes in, excites an electron in photosystem 2. That electron is shuttled down this electron transport chain, and in the process, hydrogen is moved from the stroma to the inside of the thylakoid, what's called the thylakoid lumen. That electron then goes into photosystem 1, where it is excited by light again, and that travels down another electron transport chain and creates NADPH. You might be thinking, okay, these electrons are getting used up in NADPH, so how do we get new electrons? Well, if you look here at the beginning at photosystem 2, you will see that water is split, creating oxygen and hydrogen ions. And so these hydrogen ions created here, plus the ones that are transported, create this gradient, this higher concentration of hydrogen on the inside of the thylakoid than outside. That gradient, And that electric pressure from all these positively charged ions being jammed in close to each other is going to be used when these things come rushing out of the channel protein that is ATP synthase, and they are used to create ATP. Let's take a look at this in a second way. Here is an individual thylakoid. You can see now the two photosystems, Photosystem II and Photosystem I. And notice that Photosystem II comes first and Photosystem I comes second. The reason for that is probably because this one was discovered first and this one was discovered second. So light comes in, excites an electron. The electron travels down the electron transport chain from photosystem 1. Hydrogen is pumped into the inside of the thylakoid. Meanwhile, in order to supply new electrons to this photosystem, water has been split, hydrogen ions are released into the lumen, adding to the gradient, and then oxygen is created as a waste product. That's going to diffuse back out into the environment and ultimately into the atmosphere, which we're very happy about since we are organisms that require that oxygen. Now, that electron, once it gets here, it gets shuttled into the next photosystem, Photosystem 1. It gets excited by light, and then it travels down a few more proteins, which create NADPH, which now carries the high-energy electron, as well as what we call reducing power, and you will learn more about that when you take a chemistry class. Meanwhile, the hydrogen ion gradient is used to create ATP by the membrane-bound enzyme ATP synthase. Let's just remind you that the first electron transport chain forces hydrogen ions across the thylakoid membrane. Therefore, it's another example of a hydrogen pump, just as we saw in cellular respiration. The hydrogen ions inside the thylakoid are electrically charged. They exert an electrical pressure on each other because they're mutually repulsive, and they can be used to do work by the enzyme ATP synthase, which creates ATP. Here's a list of the sequence of events that occur, and this is the level of detail you need to know about the light reactions. First, an electron in photosystem 2 gets excited by light, and it is then shuttled into the electron transport chain. Meanwhile, chlorophyll steals an electron from water to replace the one it just lost. The water splits as it donates its electrons and releases H+ into the inside of the thylakoid, and it also releases O, oxygen. The oxygens find one another fairly quickly and form the molecular oxygen O2, and then that will diffuse out of the cell. The electrons go down the first electron transport chain, pumping hydrogen ion from the outside of the thylakoid to the inside, adding to the hydrogen ion gradient. And then ATP is made by ATP synthase, and then another photon energizes the electron in the chlorophyll in photosystem 1, and that electron flows down an electron transport chain to create NADPH. Those are the events. Note that these are all happening simultaneously. This is just one way to think about it and to order it in your mind so it makes some logical sense. Now it's time to go on and look at the dark reactions where we actually make energy-rich sugar molecules. These occur in the stroma, so outside of the thylakoids, but inside the chloroplast. They require carbon dioxide, ATP, and NADPH, and they produce the precursors to glucose, ADP and NADP. So the heart of the dark reactions is the addition of a single molecule of CO2 to an existing carbon-based molecule in the stroma in a process called the Calvin cycle. The Calvin cycle is a metabolic pathway, and therefore it's driven by enzymes, and each of those enzymes has a gene, of course. For every 3 turns of the cycle, one molecule called G3P is produced, and G3P is a precursor to glucose, and it takes 2 G3Ps to make a glucose. So here's an overview of the Calvin cycle, and what I want you to note here is carbon dioxide enters, it's bonded to an existing molecule, the ATP and the NADPH are used up to carry this out, and then ultimately glucose is made. So let's look at some of the details. When the carbon dioxide is added to an existing molecule, we say that it is fixed. This process is known as carbon fixation. So that's an important piece of vocabulary that you need to know. The ATP and the NADPH are used to drive the Calvin cycle, and after 6 turns of the cycle, which means 6 carbons have been added, the equivalent of one glucose, which is a 6-carbon sugar, has been produced. And so that should make some sense to you at this point. Here are the details of the carbon cycle. That double asterisk up here means that you don't have to know a lot of the details in this diagram, but you need to understand the general process. The boxes in here are the molecules, and the arrows between the boxes refer to enzymes that cause the compounds to change from one to the other. Carbon is added to a 5-carbon molecule here. It's called ribulose bisphosphate during the first step, which is called carbon fixation. And rather than produce a 6-carbon molecule, what's strange is this produces 2 3-carbon molecules. So that's the first step of the Calvin cycle, carbon fixation. Then in the next 4 steps, we have basically the reverse of glycolysis. ATP and NADPH are used to drive these molecular conversions. And you can see at the end, G3P is produced, and that is what can be drawn out of the cycle to make glucose and other sugars. Finally, this 3-carbon molecule has to be converted back into this 5-carbon molecule. So 5 molecules of this are converted into 3 molecules of that. That's a pretty neat trick. And then carbon dioxide can be added to them. This entire diagram shows the amount of NADPH and ATP that are needed for 3 turns of the cycle. So you need to start with 3 molecules of ribulose bisphosphate, and they turn into 6 molecules of this, et cetera, et cetera. But that's the kind of detail you don't need to know. What you really need to know is that after 3 turns, you get a G3P formed, and you need 2 G3P to make a glucose. So that's 6 total turns. So that's it for today. What I've hoped you've learned is the following: that photosynthesis is the creation of glucose by using the energy in light. And the light energy, at least some of the light energy, is stored inside the glucose. In the light reactions, light energy is first captured in the form of an excited electron in chlorophyll. This electron then produces ATP and NADPH, and new electrons are supplied to the system by the splitting of water, and oxygen is released as a waste product. One of the electron transport chains in the light reactions produces a hydrogen ion gradient that is used to produce the ATP, and the other one directly produces NADPH. In the dark reactions, carbon dioxide is fixed, that means bonded to existing carbon molecules, and ATP and NADPH are used to then turn those molecules into glucose. The light reactions occur in the thylakoid membranes, and the dark reactions occur in the stroma of the chloroplast. And one thing I neglected to mention, but I'll say now, is that the light reactions do indeed require light to occur. But the dark reactions do not require darkness, but they can happen in the dark if ATP and NADPH is still available. So that's a lot to master today. So after you've studied a little bit more, go outside, look at nature, ask some biological questions, and bring them back to class. And I'll see you soon.